Good day, everyone. My name is Michael Ray. I am science writer at GlycoSENS Research Foundation, and I'm joined today by Dr. Jonathan Clark. Dr. Clark is the head of biological chemistry at the Babraham Institute in Cambridge, UK. So Jonathan, you've been working on a really massive, detailed project on the extracellular matrix with GlycoSENS Research Foundation for a number of years now. Big picture, what's the problem you're ultimately trying to help us solve?
Okay, in a nutshell, what we're looking at is why chemically the extracellular matrix changes with age.
And so let's start with what is the extracellular matrix?
So in a nutshell, the big picture is that we are trying to identify the changes which occur in the extracellular matrix. That is the material that surrounds and supports cells within tissues. Specifically, we're looking at two proteins, collagen and elastin, which make up a large percentage of the protein content of the extracellular matrix. And the thinking is that if we can identify the changes which occur with age and identify what makes young tissue behave the way it does, that we might be able to find ways of treating tissue so that it behaves more like younger tissue. So basically reversing some of the chemical changes that we see with age.
Right. And now, what are some of the sort of known changes in the ECM at the level of functional impacts? Like what kind of age-related diseases and disorders and cellular dysfunctions do we see when the ECM goes awry for reasons that are not yet clear?
Okay, so it is very obvious to everybody when you see an old person in the street, you know that they're an old person. And if you see a young person, you know they're a young person. And equally, actually, when you see old pets, it's very easy to tell that they have aged. So what is it about that? What are the cues that you're getting? Now, in terms of medical impact, though the cues that we're picking up unconsciously actually can have quite important effects in an elderly person. So you would find things like thinning of the skin and less elasticity. You can find in tendons, they're more fragile and more susceptible to breakage. So it's very difficult to say that there's any one thing that creates a universal disease. There are lots of different disease states which are down to the fact that the extracellular matrix isn't functioning properly because of the changes that have occurred with age. So in terms of a parallel everyday effect that you might see in materials around the house. If you think about the plastics in the bucket or the rubber in a windscreen wiper, that ages because the chemistry is changing over time, and a bucket can become more brittle and a windscreen wiper can become less flexible. And those are sort of parallel examples of the sort of changes that are occurring within the ECM that can lead to similar difficulties with the way tissues and organs function.
And so the specific form of alteration in the ECM that you're researching with GlycoSENS Research Foundation support that's driving a lot of those mechanical changes is a chemical modification. Tell us about that.
That's correct. The collagen molecules and elastin molecules can link together, and we call the linking cross-linking. And there are a range of different chemistries that can be involved, so that gives you a range of different types of cross-link. And within our research area, we classify the different types of crosslink that you can get as reversible and irreversible. Now, you can think of the reversible ones as being a bit like Velcro. They can form, break, and then reform again very easily. Whereas the irreversible ones, once they've broken, they can't really form again, but it takes a lot more energy to actually break an irreversible crosslink. But what you find with age is that in young tissue, there tends to be a very high level of reversible crosslinks. And then as the animal or the person ages, you get fewer and fewer of the reversible crosslinks. And the position of the reversible crosslinks can change into irreversible crosslinks. And overall, you actually see a drop in crosslinking, even though there is an increase in the irreversible crosslinks.
Right. So you're seeing is not only in the total number of crosslinks, but in the kinds of crosslinks. But the net result is that there's an overall reduction in overall cross-linking, even though some specific kinds of cross-links are increasing quantitatively with age.
That's correct. It's historically in this research area, people have been very focused on the fact that irreversible crosslinks increase with age. However, in our research, what we've highlighted is that many of the characteristics or properties of the reversible the reversible crosslinks are actually ones that you want in a tissue that's going to behave like a younger tissue. So what do the reversible crosslinks actually bring to the party? Well, what they allow is a stretchiness and an adaptability which is lost in, and a repair function, because they can reform, which is lost in older tissues. So although the irreversible crosslinks that you get in older tissues allow much greater force to be exerted on that tissue, once something goes awry, there's little ability for the tissue to adapt or absorb the energy that's being applied to it. So my feeling is very much that if you want to rejuvenate tissue, you not only want to decrease the irreversible crosslinks to some extent, but you want to increase the reversible ones. And I think that's very important, because I think a lot of the characteristics that people are expecting to get from just removing the irreversible crosslinks won't happen if they haven't also increased the reversible crosslinks. Now, in a minute, I must stress that is hypothesis, but that is a hypothesis that we are actively pursuing. So, yes.
Yeah, and actually you've taken that so far as to think that perhaps if you were to remove some of the excessive crosslinks of some particular classes without replacing some of the crosslinks that are lost with age, that you might actually have the tissue literally fall apart, because these fibrils that are supposed to be connected together by crosslinking are no longer bound by these age-related irreversible crosslinks, but are also not bound by these more youthful reversible crosslinks. And so you could wind up causing as much harm as good if you simply break the age-related crosslinks.
That's correct, because the situation that we would be heading towards, based on the research that we've done, would be more like that is found in diabetic tissue, where actually you see a decrease in the reversible crosslinks because the raised glucose levels in diabetics actually interferes with the reversible crosslink formation. But particularly in mice, you don't, in the time frame of diabetes that we've looked at, you haven't also had the increase in irreversible crosslinks occurring. And in those in diabetics, what you tend to find are breakages that you wouldn't normally find in, say, things like tendons. And that's, that sort of supports the idea, the hypothesis that we've got, that just reducing the number of crosslinks is going to be a problem. And actually, when you think about it intellectually, if you have a lot of little strings all joined together and you remove all the linkages between them, they are going to become much less intact.
Yeah, yeah. All right, so that has major implications. That's important. Now, we've been talking mostly about the aorta, and most of the work that you have done over the last several years has been with tendon, but in fact, you're now moving on to the aorta, which is a really important tissue that we've focused on in the past. Tell us about your work in the aorta.
That's correct. The aorta is a very complex organ. It seems very simple when you first look at it because it's a pipe. It's a tube that comes out of the heart. But actually, the way it works is very, very important. It acts like a balloon. So when the heart pumps blood out of it, that balloon fills up, and then when the valves on the heart close, it gradually contracts while the heart is refilling. And what that does, it does two things. One is maintain a smooth flow of blood around the body, but it also removes the pressure wave that you would get if that expansion, that balloon effect, couldn't happen. Now, it is well known and has been shown that the aorta stiffens with age. And the implication of that is the ballooning effect is much harder to achieve. And so when the pressure wave comes out of the heart, as the heart pushes the blood out, it doesn't expand as much. What that does is it creates a situation where the blood flow around the body is more pulsatile, and it's at there's a higher force behind that pulse. And that's potentially very damaging to peripheral organs. The other feature that occurs if the aorta can't expand is the heart has to work much harder to push the blood out of it. And that's also detrimental to the heart. So you've got a problem on both sides of that aortic problem of stiffening.
Right. And just to spell it out, I mean, the organs that you're talking about being hit more directly by the force of the pulsatile action of the pulse, we're talking about organs like the kidney and the brain. And of course, that's already, we know, driving strokes and renal disease with age. And so potentially, at least, if you could normalize the mechanical properties of the aorta, you would be reducing the amount of damage you are delivering to the liver, to the kidneys, and to the brain, and to other end-target organs. Okay.
And bear in mind that these other organs are also arterial in nature. You know, the blood vessels there are also aging and becoming more fragile and more susceptible to damage. And so, you know, the combination of the more pulsatile nature and higher pressures is also going to have more of an effect on an older organ than a younger one.
Right.
Yes.
And you've identified a couple of sort of complicating factors in the research to date, one of which is seeming contradictions in reports on what's actually happening to the mechanical properties of the aorta. So talk about that.
And this is very common in the field of collagen research in particular. You know, it's a very well-established field that's people have been researching for 40 or 50 years, and things creep into it that are very hard to explain and sometimes just wrong. But in terms of the aorta, you can find studies where people have actually decreased the number of reversible crosslinks and seen a stiffening. And there are other studies where they've decreased the number of crosslinks and seen the opposite to stiffening. Help me out.
Restoration of elasticity.
less stiff.
Yeah.
So yes, there basically you can find research where if you decrease the cross-linking within the aorta, you see opposite effects. It can make it stiffer or less stiff.
Right.
And that's difficult just from reading the papers to understand. You actually have to see what's going on. And some of the issues there are that the stiffening of the aorta is probably due to a number of different effects, and cross-linking is one of the effects that's important. But there are almost certainly other things going on, and we need to try and understand how much of the effect is actually down to cross-linking, and whether reversing or changing the profile of the cross-linking would be beneficial or not.
Right, right. And to make the whole field even more difficult, we have this problem where the profile of collagen, the actual like collagen protein types, varies not only between tissues, but is different between mice and humans.
That's correct. The cross-linking is dependent on the organ that you're looking at. And even within a particular tissue type, say tendon, if you look at a flexor tendon or an extensor tendon or the Achilles heel and the palmaris longus, which is a tendon which runs up the hand and then spreads out across the hand, and in apes is for gripping onto branches and things. The functions there are quite different, and that actually seems to lead to different cross-linking profiles. Now, nobody really understands how those different cross-link profiles are controlled biologically, or it could be the mechanical action itself which is driving those differences. But there are clearly differences. Sorry. And on top of that, between, say, mice and humans, there are small differences in the distribution of enzymes that cause cross-link changes. There are small differences in amino acid sequence, which can have an impact on the type of cross-linking formed. And so it's important not only to study the type of cross-linking in different tissues and in different locations, but also to look at the difference between the cross-linking in mice and the cross-linking in people. And a lot of the fundamentals can be done very easily in mice, and that's why we work with mice a lot, because you can look at the fundamentals of aging and the changes in collagen over 2 years, but you do need to do the translation into people and look at the similarities and the differences. And we are just starting to move into looking at human samples and understand the similarities and differences there. So that's very exciting, actually. And already I can tell you it's opening up interesting changes that are almost certainly occurring in mice, but they're a lot harder to detect in mice because the amount of tissue and the speed at which things are changing, the situation is different. And I think, for instance, we are probably seeing intermediates in the synthesis of glucosapane, biological formation of glucosapane, for example, which theoretically you know must be there because we can find it in mice, the final glucosapane molecule, but we can't see the things that lead up to that final molecule. But in some of the human tissue, I think we have seen it. And the reason why that might be important in this context is some of these intermediates might be easier to break in the long term, because a final glucosapane molecule is a very rigid, chemically stable molecule. But some of these intermediates, may be much easier to actually tackle. So I think that's very exciting.
Yeah, yeah, all good stuff. We've talked about the presence of both normal physiological cross-linking and abnormal cross-linking that was not necessarily, designed by the organism to be there and accumulates over age. And the blood sugar, glucose, plays a big role in that. Talk about that and what the implications for that are.
Right, so what we found is the glucose is actually, the circulating blood glucose is an important factor in the change and development of cross-linking within collagen. So the circulating glucose can react with collagen, and it reacts at the sites where the reversible crosslinks are formed. So in doing so, it can actually block the formation of reversible crosslinks. If it sits there long enough, it can actually then develop into the sort of molecules that people have heard about before, the advanced glycation end products. They're things like GlycoSENS. But one of the interesting things that we found in our research is that even if it doesn't go on to form those irreversible crosslinks, the binding of glucose to collagen itself can cause a stiffening. And presumably that's either because the glucose forms hydrogen bonds within the collagen network, or it actually physically stops rotation movement of the collagen molecules, and so they become stiffer. A very interesting result that we got when we started testing stretching tendon in vitro was that this added glucose is actually able to come off the molecule, off collagen, when you stretch it. And so there is a degree of reversibility in this process that is stimulated by stretching or movement of tendons. And I think that potentially is of very high importance to people, because I think that it says that if you do exercise or stretching, that is possibly one thing which is going on and could be beneficial that we've been unaware of before. One of the ideas that is going through my head at the minute is if you had a treatment where you wanted to increase the number of reversible crosslinks, would you sensibly tie it in with some sort of stretching exercises as well during treatment? Because maybe that would improve the efficacy of what you're trying to, the materials that you're trying to administer.
And to spell that out, the idea there would be you would break these temporary crosslinks and expose the binding sites where these reversible crosslinks would be reformed by the hypothetical rejuvenation biotechnology.
Yes, and it would be something like lysyl oxidase that would be converting lysines that are there to a form that can form crosslinks.
Right.
So, and that is leading on to some very interesting research that we're currently trying to achieve, which is we're asking the question, if you were to introduce these reversible crosslinks into older tissue, would you actually be able to get some of the characteristics of younger tissue without actually having to remove some of the age crosslinks? And for us, that's quite a different approach to the approach that everybody else is taking at the minute, which is to just try and break the irreversible crosslinks like glucosamine. And if we're successful, you could imagine that both technologies could be combined to rejuvenate tissue even more effectively. I don't know, was that?
Yeah, no, that's good.
Good
okay.
enough.
Now, we talked earlier, we've been mostly talking throughout this discussion about the sort of mechanical properties of the tissue as if it was a windshield wiper or a bucket. But the ECM actually also provides a kind of signaling system that gives feedback to cells in our tissues. So talk about how the implications of alterations in the ECM with aging interface with other rejuvenation biotechnologies. And I'm thinking most especially about cell therapies.
Yes, well, this is something which has been of interest and worried me to some extent for a long while. There's an increasing body of evidence that shows that cells within the ECM are responding directly to mechanical signals that they're receiving. And so if you take old cells and put them into younger tissue, they behave more like young cells. Equally, if you take young cells and put them into a stiffer tissue, they behave more like old cells. And so this raises a sort of chicken and the egg type question. You know, if you're going to take a population of cells and rejuvenate them, and then put them into old tissue, are they then going to just behave like old cells? Do you have to have rejuvenated the extracellular matrix first before you put your rejuvenated cells in? Or will you be able to inhibit the signals that the cells get from the matrix so that the extracellular matrix gets replaced before the rejuvenated cells have realized that they're in an old extracellular matrix?
Right.
So, you know, this is an area that we would quite like to work in ourselves, where we actually take some old cells and young cells, switch them around into decellularized tissues, reculture them, and modify the characteristics of the tissues that they're in, and see whether we can actually change the way the cells are behaving in, say, tendons. Because a lot of this work has been done in quite different systems. So when we talk about old tissue, sometimes they're not actually working with what you and I might call old tissue. They're working with tissue from animals that are old by their standards, which might be 16 weeks. So they've waited 32 weeks to get an old animal. But in my case, I would view that as actually a fairly young adult animal. I wouldn't actually see that as an old animal.
Yeah.
And that is not uncommon in, or has in previous literature been quite a common problem, that people that claim they're working with old tissue or old cells are not really working with what you and I would call old ourselves.
Yeah.
They're really just young adults, and they're not juveniles.
That's been a problem throughout the whole area of aging research. Well, people will often say, well, this is an old animal, and it's really just an adult animal, and the young animals you've got are, you know, barely weaned.
Yeah.
But
well,
to
doing
that,
this sort, sorry,
point,
doing this. So yeah, what I was just going to say was doing this sort of research requires planning, because if you're truly going to work with old animals, you've got to think 2 years ahead.
Yeah, because you've got to have 2-year-old mice, right?
Yes.
Yeah. Okay, so this is all very exciting work. We're talking about how we could prevent disastrous outcomes like stroke and kidney disease. We're talking about how we could restore the youthful function of tendons and other mechanical tissues. And we're even talking about how to make cell therapies work as they are intended, as opposed to putting, as it were, new wine in old wineskins. So excellent stuff, a lot of work to do. Good luck with it, and let's get results as fast as we can.
Thank you very much. And look forward to speaking to you in the future about our ongoing research. It'll be excellent. Yeah.